So just to launch into the talk, I think the title is pretty self-explanatory in terms of just sort of the research journey that I've been on to try and understand two very important aspects of evolutionary biology, particularly in humans, and that is reproduction and aging. And it's been known for quite a while that the two act hand in hand, so to speak, and they both affect each other in very deep ways. So, one of the things very quickly, just in terms of giving a very, very short background to all this, there are a lot of wonderful ideas in terms of the evolution of aging and how we understand aging, going way, way back. And I'm not doing justice to this, but just to choose some very choice and important landmarks in our understanding, our scientific understanding of aging. We can start with Raymond Pearl talking about the rate of living and how metabolic processes can engage and influence how fast we age and how fast we degrade with time. The other idea that came about by J.C. Williams in 1957 was one about antagonistic pleiotropy. Very simply, that means that traits will affect each other. That if one trait is influenced by natural selection, it's very likely to affect the expression of other traits. And in this case, antagonistic pleiotropy means that when you select for one trait, let's say for a greater reproductive output, that it'll have a cost someplace else. Um, and then fast forwarding down to the disposable soma theory, uh, we could talk all day about the disposable soma hypothesis, but in general, it states that reproductive effort, how much time and energy that goes into reproduction, can affect the rate of aging in organisms and how fast they degrade. And then finally, I just wanted to give a shout out to Arlene Geronimus, who's actually not a biologist, but actually a cultural anthropologist, who
Postaram się jakoś
um,
skontaktować.
made a very astute observation that she called weathering,
No dobra, no.
and that is that uh, females who engaged in hardship or endured hardship. In her case, what she's talking about things like racism or
Co byś chciał?
very adverse
Nie,
socioeconomic.
nie.
And again, that just weathering is a process by which other social factors, environmental factors, can influence aging and physical degradation, particularly in females. So these are all sort of things that kind of inform this research. And my own interest in this actually goes back to my days when I was working with the Ache people in Paraguay. And I'll bring attention to this young woman here. Her name is Marilyn Becakramagi. And don't worry, there won't be an Ache language test today. Um, but Becakramagi, I knew her since she was about maybe about 7 or 8 years old. It's hard to tell. But by this time, at this point in my research, she's already a grown woman. She already has, these aren't all of her kids. I think three of them are her kids. Um, but if you were to look at her and you were to do an assessment in terms of guessing how old she is, you may guess she's in her mid 30s, perhaps late 30s, when in fact, in this photo, she's only about 17. And it really brought home the point that reproduction, as well as the hard life that they have, those are things that are very difficult to disentangle, um, can have an effect on your physical degradation that we could call aging. So these are sort of the things that kind of motivated this research to try and understand it. The other is many years ago, while I was asked at Yale to give a talk to the staff, sort of the clerical staff, at a lunchtime talk. And I was just starting to talk about this and get interested in it. And I told this group of lunchtime clerical workers that I was very interested in how perhaps children can make you age faster. And very politely and very respectfully, they all burst out in laughter, said, "Oh, silly professor, I could have told you that." So it was already sort of well established that aging and reproduction kind of go together. So what are the sources of aging? Well, the goals of this particular body of research are to identify sources of aging. The other is to develop ways to measure the costs of reproductive effort and trade-offs. Now, this is a very tricky thing. It's a very difficult thing to do in any organism, and it's especially difficult in humans. Um, Years ago, very prominent, I won't say who it was, professor said, well, why don't you just do X, Y, and Z? And I said, well, professor, I can't do that because unlike you who work with Drosophila, I cannot grind up my subjects and measure their DNA. So it's very, very difficult to do this in humans. But we've developed methods in order to do this, to actually test these predictions. And then finally, one of my tangential interests is how this research can contribute to what I call informed reproductive health. So, very quickly, what are the costs of reproduction in human females? Well, obviously, gestation pregnancies are metabolically and energetically very expensive. Here, throughout the course of gestation, you can see how basometabolic rate, which is the amount of calories over a period of 24 hours, rises throughout the pregnancy and then finally terminates at birth. But you can tell how BMR can rise up to about 400 extra calories um, during pregnancy. So that is one cost. The other cost, obviously, that's often underappreciated is the cost of lactation. With the cost of lactation per day with exclusive breastfeeding is about 500 calories a day. Now, there's a lot of variation here. There's variation in terms of the intensity of breastfeeding, the energetic status of the mother. There's a lot of the residuals around this number can be quite large, but it's pretty safe to assume that you can start with sort of a ballpark estimate about 500 calories per day that is necessary to support lactation. And being the hamburger loving American that I am, that amounts to about one Big Mac, one large McDonald's hamburger per day. So clearly, there are a lot of metabolic costs that go along with reproduction in females. However, are there other costs in addition to increased energetic demands of pregnancy and lactation? Are there other costs associated with reproduction in females? That's going to be the central focus here. And I'm delighted that Grajna is here because this particular paper has influenced me tremendously, and my graduate students know this graph like the back of their hands because I show it so often. I also use it in my undergraduate course. Um, Here in this particular study done by Dr. Grażyńska and her colleagues, we look to see uh, if there's any association between lifespan and offspring, in this case, daughters and sons. And I'm going to focus down here on maternal lifespan, and as you can see, there is a very, very prominent sort of dose-dependent response in terms of the effects of number of daughters and number of sons on lifespan. So with one daughter, lifespan is expected to be about 78, and these are in rural Polish women, and the same here. And if we go down here, maybe even to, let's say here where the residuals aren't so large, we're looking at about an 8-year difference in lifespan. Now, we'll come back to this because other studies have not found this effect. But Dr. Jasinska has made a very, very compelling argument as to why that is, and we'll get to that in just a bit. But this shows that demographically, there's some very compelling evidence to show that reproduction does have a cost in terms of in terms of lifespan. I wish I had time to talk about dads here, but as you can see here, there is no effect And in fact, it actually goes up when you have more daughters. And I think this is a good example of anthropology at work, where social and biological factors sort of come hand in hand. But for now, um, you can see how reproductive effort in terms of the number of offspring can shorten lifespan. The question is why? What is the mechanism? Well, one of the basic costs of reproduction is oxygen consumption. And when you look at oxygen consumption during pregnancy, you can see that it goes up quite dramatically. Particularly, most of that oxygen goes to fetal growth as well as many of the tissues that are associated with reproduction. Um, this increase in oxygen consumption comes with a cost. It's not just the actual cost of increasing your BMR and your respiratory function and all the things that go along with that, but aerobic metabolism incurs another cost that is going to be central to this talk. That cost is oxidative stress. Now, the biochemistry of oxidative stress is complicated. It's very involved. But in short, one can sort of think about it as the toxic byproducts of being an organism that relies on oxygen for metabolism. Um, the evolution, if you will, of aerobic metabolism was very important for air-breathing organisms such as ourselves and obviously our primordial ancestors. It made us very much more efficient at transforming calories into energy, but it came at a cost. And that cost is during the process of aerobic metabolism, incorporating oxygen as opposed to anaerobic metabolism, it creates toxic byproducts. And those toxic byproducts, and this is one example of one, 8-OHDG, which we'll be talking about, can do serious damage to tissue, cells, and DNA. And we know, and it's quite well accepted, that oxidative stress is a major contributor to physical degradation that we associate with aging. It's not the only one. There are many, many other things that are involved, but oxidative stress is a very, very central player in the aging process. So now we have a clue that if oxidative stress is central to aging, and oxidative stress may increase with increased aerobic metabolism during pregnancy, then maybe there's an association. There's a link here that's worth exploring. So very quickly, some of the early studies on oxidative stress had to do with smoking. If you're a smoker, you generate a lot of oxidative stress, both in terms of the toxins that are associated with the smoke itself, as well as changes in aerobic metabolism. And this is a famous study, a sort of a chimera between um, a set of identical twins. One is a smoker over here, twin B. The over here, twin A is a non-smoker, and you can see just in the phenotypic differences in their face that smoking accelerates some of the processes that are indicative of aging. And the evidence is pretty clear if you look at, again, our friend 8-OHDG. You can see how 8-OHDG is much higher here in these black bars of smokers compared to non-smokers. However, moving beyond smoking, it's also well established that during pregnancy, there is the possibility of increased oxidative stress as a result of increased aerobic metabolism during pregnancy. And we won't get to a chance to hold this. As a wonderful graph by, again, a Polish research group. And here, what they're showing is all of these different factors associated with um, uh, what are called reactive oxidative species. Those are the actual toxins that actually cause damage. And when those toxins, those reactive oxidative species, are released, and if they are not dealt with or soaked up by protective factors, it can cause oxidative stress. And if it's very intense, it can cause damage and health challenges both to the fetus and to the pregnant women. So there's very compelling evidence that oxidative stress is something worth looking at. Now, uh, this slide is meant primarily for myself. I made this slide early on to try and figure out how do I actually put this in terms that people can actually understand and try to get their brain around. Um, if we look at a life shortening factor and compare it with the cost of reproduction. So in this case, there's a very famous study called Time for a Smoke. One cigarette reduces your life by 11 minutes. And it's how smoking is bad for your health and actually shorten your life. Now, if we draw on the number, just as an example number from Dr. Jasinska's study, that each child costs a woman about 95 weeks of life, this is the equivalent of a woman smoking 1.5 packs of cigarettes per day for 10 years. That is what one child is equivalent to. And if you sort of extrapolate that to 30 years, sort of the reproductive lifespan of a woman, it works out to about 1/2 pack of cigarettes a day. Um, and the reason I do this letter, this small exercise, it's to sort of bring home the impact of how reproduction can have a cost on aging and well-being. Now, this idea isn't really new. It's been well established and it's been discussed for many years, but within non-human animal models. So this is a graph generated by Speakman, a very prominent metabolic physiologist. And what he shows here is that when you look at a non-breeding animal, in this case, they've looked at rodents, birds, what have you, and you see energy here, and you see a lot of that energy goes to somatic protection, it goes to metabolism. These are reactive oxidative species, which cause oxidative stress. It causes damage and it affects survival. He breaks this out in terms of if you contrast this with a reproducing animal, and you look at energy and you see how much energy actually goes to reproduction and how reproduction can contribute to reactive oxidative species and oxidative stress and actually increase the damage that is done by oxidative stress compared to a non-breeding state. So these are sort of a simple model that he developed that we sort of built on to develop our understanding. And when you kind of look at this and sort of bring this all together for human models, we end up with some really interesting possibilities. In this case, this is a graph developed by Peter Ellison and myself. And here we have in these spikes, we've got sort of a very basic model of human female reproduction. What we're calling somatic reproductive effort. Here's age. And in this case, we got 1, 2, 3, 4, 5 bouts of reproduction, 5 bouts of pregnancy and lactation. There is a recovery period. But in each one of these bouts of reproduction, there is also an increased bout of oxidative stress, potential increase in bout of oxidative stress. And so one of the predictions that we have is that the more bouts of pregnancy and lactation that you have, the greater exposure you have to oxidative stress, and that could contribute to accelerated aging. You'll notice here that in males, it's a very different model. That's a whole different talk. I debated which talk to give. I chose to give this one instead, but I'll mention this a little bit later in terms of how males kind of fit into this picture. So how do we test this? Thanks to our wonderful collaborations with Grażyna and Anna Zemkiewicz and others, we decided to look at this in the Mojalika study site from which the samples were drawn and the data was drawn to look at whether or not oxidative stress was associated with gravidity in terms of the number of pregnancies and other aspects of reproductive effort. And given the audience here, I'm sure some of you are already very familiar with this. It was a very comprehensive study looking at many, many aspects of reproductive effort as well as various biomarkers. In this case, we looked at post-reproductive women, so postmenopausal women, and primarily one of the benefits of this is that it helped to screen out the effects of or the hazards of childbirth. And so the hypothesis was that in this population, gravidity, the amount of energy you put into pregnancies or in reproductive effort in and of itself, should be associated with biomarkers of oxidative stress. And so we concentrated on three biomarkers of oxidative stress. One is called 8-OHDG. 8-OHDG is the repair outcome of reactive oxidative species forming a lesion on DNA. Enzymatic factors cut that out, package it, and it gets excreted in urine. And it's a very, very useful biomarker of oxidative stress because it shows you DNA damage on a one-to-one basis in terms of the number of molecules that you actually measure in urine and the amount of damage in DNA. The other biomarker we looked at was called SOD. It stands for superoxide dismutase, and that's a protective factor. The other one was T-bars, which didn't work out too well, but we ran it anyway. And these are the results. We published this in 2016 in PLOS One. And to our knowledge, this is the first, if not one of the first, published pieces of evidence showing a proximate physiological mechanism that shows an association between reproductive effort and aging in humans. It's been done in non-human animal organisms, animal models, but not in humans. So here, when you divide the group into low and high gravidity groups, we look at 8-OHDG over here in the blue, and there is about a 20% difference. Those in the high gravidity group had 8-OHDG levels that were about 20% higher compared to the low gravidity group. When you look at SOD, there are various types of SOD. That's why you see this copper zinc, but we'll just call it SOD for the moment. SOD, there's also a difference of about 20% between the high and low gravidity. The T-bars, which is a measure of what's called peroxidative stress, there was no effect there. There was no difference. But what this shows here is that in postmenopausal women with high gravidity, there is a persistent echo of increased oxidative stress in association with reproductive effort, as reflected by more pregnancies. So this was a very, very compelling study that we thought was very important for addressing this question. A few years later, I forgot to mention that was done at the Mojalika, well, maybe I did, with the Mojalika field site communities. A few years later, in conjunction with Amelia Sansilio, who was a PhD student at Yale at the time, as well as a number of other collaborators, Dr. Sansilio basically replicated this study in a more sort of comparing rural with urban environments. Again, looking at populations at the Mojalika field site, as well as in Krakow as an urban environment. And what she found was basically the same thing as indicated by the arrow here. When you look at 8-OHDG, 8-OHDG was positively associated with gravidity, as was SOD. And interestingly enough, there really was no difference between the rural or urban environments. We also replicated this study in the United States, looking at women within the state of Connecticut. These are women who are basically urbanized, sedentary, working women, to see if we would see the same effect. And the reason we did this was to follow up on some of the predictions of Professor Jazienska in terms of whether or not energetic status and ecological context will have an effect on this potential trade-off that we're seeing in Poland. And what we found in the United States was that there was no effect. There was actually no difference in A2HDG between nulliparous and parous women, and between those with live births, there was no difference in A2HDG. So what this suggests is that ecological context matters, energetic status matters, and that's one of the things, as Professor Jazienska has argued in the past. This is probably one of the primary reasons why there are disparate results between many studies. How some studies find a trade-off between reproductive effort and measures of aging, and others do not. Many of those studies do not take ecological context or energetic status into consideration. And that is an absolutely important factor to consider, given the fact that even from if you're, and it's something it's a little bit of a pet peeve for myself when I talk to my graduate students. When researchers try to find trade-offs, they forget one of the basic predictions or conditions that need to be met, and that is a key resource needs to be limited. When the key resource is not limited, you're very unlikely to see a trade-off. So you have to be aware of that. And in this case, again, ecological context and energetic status are very important. And so now we can go back to Speaksman's model and now have a more complex model that actually breaks out two things. One is the tissues involved, blood, muscle, liver, kidney, brain, others. And over here in a reproducing animal, you're actually breaking out those tissues. You're actually considering energetic status, and you're also bringing in reproductive hormones, which are actually going to be important when we look at this question in males in just a second. So clearly, what we've done so far in females is to test a very basic model, but there's more work to be done in terms of identifying other factors that are very important. Before I move on to the males, are there any questions on any of this? If not, I can wait to the end. Very good. We'll go on to males then. All right. For a lot of my, just to pivot a little bit, for a lot of my research career, I've looked at the reproductive ecology of human males, and I've continued this in a ongoing collaborative project in Ecuador with an indigenous community known as the Shuar. The Shuar live, excuse me, in the south, well, actually the north, no, actually the southeast portion of the country along the Peruvian border. They're an Amazonian population. The groups that we look at are still a foraging population, still relying much on hunting and fishing. But a lot of the project has to do with looking at the effects of their transition to a more market economy, sort of wage labor and engaging in a capitalistic economy with nearby towns. So this is just what I'm going to talk about today is just one facet of that overall project. And in looking at males, the model that I kind of is my touchstone that I kind of rely on is this early model that I developed going on almost 25 years ago. I choke up to think about that, but it is almost 25 years ago. And that is that male energetic allocation is very different compared to females when you're looking at reproduction. Males do not lactate, at least not commonly. They do not get pregnant, at least not commonly. And when I'm talking about males, I'm talking about the biological definition of male. Um, and in males, you have energy availability with some tax on food, but all that energy goes to somatic investment. You don't have to share it with a growing fetus. Again, commonly no lactation, but there are still allocation decisions to be made between survivability, tissues that can increase survivability, such as fat deposition in lean times, but reproductive effort in the forms of sexually dimorphic tissue and our old friend testosterone, which supports a lot of these aspects of male reproductive effort. So it's important to sort of ground yourself that the dynamics are going to be very different in many ways compared to females. And if you look at this is a really nice graph from Herman Ponzer from this amazing study he published a few years ago in Science, sort of compiling measures of total energy expenditure between males and females. And one of the things you can see is that total energy expenditure, which is primarily a function of sexually dimorphic tissue, there are differences in activity and other things, but body composition is very, very different. And in this case, that body composition results in about um, maybe anywhere from, depending on who you ask, 10 to 15% difference in basometabolic rates just standing still. That differences in body composition, greater muscle mass, which is one of the primary contributor to basometabolic rate, is higher in males and persistently higher throughout their entire life. So that's something to consider in terms of how we think about reproductive effort in males compared to females. So what does this all have to do with oxidative stress? Well, again, most of the research that has addressed reproductive effort and oxidative stress in males has been in non-human animals, for example, in birds. And this is one classic study where um, they treated these little finches with either an anti-androgen, which can suppress testosterone, a control, or males with testosterone implants. Now, what this is, lower numbers are better health-wise, if you will. So these are resistance to oxidative stress. Uh, the more resistant you are to oxidative stress, the less likely you are to have oxidative stress damage. And here, when you block testosterone, these birds were much more resistant to oxidative stress compared to those that were treated with testosterone. There was a huge difference. And it results in a lot of tissue damage and DNA damage that can come about down the road. Um, again, humans aren't birds, but we can try and sort of look at this in humans in different ways. And this is where the Shuar come in. One of the uh, really interesting studies that we've been doing has been in collaboration with Herman Ponzer and looking at metabolism. And so one of the things that just at a basic science level that we're very interested in is variation in basometabolic rate. So with the Schwarr, what we did was we provided them with doubly labeled water. And I don't have time to get into it, those you're probably familiar with it. Um, Measures of metabolism, usually doubly labeled water, and then measuring output through urine is pretty much the gold standard of measuring basometabolic rate, the amount of energy that individuals are burning at any given time. Um, and so what we're able to do is actually measure basometabolic rate among the Schwarr because what Herman and others have shown is that there's a lot of variation in BMR between populations. And so what we found, which was quite remarkable, that we published about 6 years ago, was that the Schwarr have among the highest levels of metabolic output and energy expenditure of any population in the world. Um, if I can this is probably the most telling study here or a figure. Here are the Shuar in black, and these are various other populations. Here's US, I believe South Africa, I believe Ghana, and a few others. Um, and you can see the Shuar are much, much higher, especially over here with males. Um, and so if we were to see any aspect of oxidative stress, um, it would be among the Shuar because they have such high metabolic outputs and their aerobic capacity is so high. So we decided to conduct a study to see whether or not oxidative stress is associated with reproductive effort in Shuar males. And we have another study going on with Shuar females. We're still compiling that data, but stay tuned. Maybe we'll have that published by the end of the year. But for now, we're sticking with males. And so since males don't gestate or lactate, we use testosterone and anthropometric measures as a measure of reproductive effort. And what we basically predicted, this is the model that we're kind of looking at, is that testosterone is reflective of reproductive effort. It promotes anabolism and metabolism, anabolism being the building of sexually dimorphic tissue, particularly muscle. It can increase metabolic rate, but as you increase metabolic rates, it has the potential of increasing oxidative stress. And that oxidative stress should result in increases in 8-OHDG and SOD. And then we have just getting older, aging as one of the factors that sort of plays into all this. So, what did we find? What we found was that testosterone had absolutely no effect on any of these measures of oxidative stress. So when you look at 8-OHDG, in the total model, when you look at all these different factors, there's really nothing there. When you look at the most significant model and you focus on these three variables, SOD, age, and testosterone, um, only SOD and age were significant factors in predicting this model of 8-OHDG, which is what you'd expect. As you get older, you build up 8-OHDG. SOD goes up with, I mean, SOD goes up with 8-OHDG, so that's not a surprise. But testosterone over here, you see, had absolutely no effect. And when you look at SOD, the exact same thing, not surprisingly. So what happened? Why do we see this effect in rural Polish women and urban Polish women, but not among Shuar males? Well, there's a number of things that we can sort of point at. One is perhaps there are other oxidative stress biomarkers we should be looking at. There are many others that we could be looking at. I don't think that's really the case because 8-OHDG and SOD are really predictive, but something to consider. Limited sample sizes. We had about, depending on the variable, anywhere from 30 to 50 to 60 individuals. So perhaps. There are other hormones maybe we should have measured, like cortisol, but I'm not too convinced of that. It was a cross-sectional study, which has its inherent limitations. But one of the things that I think is really important is that there are other measures of male reproductive effort that we should consider. And perhaps another thing that we need to consider is that based on some of my previous research and the research of many other colleagues, testosterone levels tend to be very, very low compared to urban, let's say, urban European, urban American males, um, and show a lot less variation. So perhaps in this case, testosterone is not going to be a viable biomarker of reproductive effort, at least variation within males in association with oxidative stress. So just to give you a few examples of this in terms of how we're sort of rethinking um, reproductive effort in not just human males, but primate males. Um, One thing that we need to keep in mind is that reproductive effort in males, human males, is much more variable compared to other primates, especially great apes. Um, Human males are the only great ape that have the potential of exhibiting any significant parental care. Um, Chimpanzees do not do it, orangutans don't do it, gorillas don't do it. There are some exceptions in other primates, such as these callitrichids in South America, where males will carry their offspring around on their back to alleviate some of the burden on the moms for an extended period of time. And this can be, at least in this species, a considerable expression of reproductive effort. We actually have a graduate student here at Yale, David Wood, who has put little accelerometers on these guys in captivity and has been measuring their energetic output when they have an infant on the back and when they do not. So that's something something to consider. The other things they have to do with paternal care are not only the energetics, but other aspects of paternal care that we need to consider. Um, Foraging effort is something that some anthropologists have argued as being an aspect of reproductive effort. But you have to sort of be careful here because one of the bits of wisdom that I received from Peter Elson many years ago was, well, everything is reproductive effort, right? And it's very tricky to start determining where those boundaries are. But I think since foraging and hunting is very important in humans, it's something that I think is reasonable to consider. So, where does this all leave us? Well, um, I think for females, I think it's safe to say that reproductive effort is a source of accelerated aging in human females. I think it's safe to say that greater oxidative stress in response to reproductive effort in females is a likely source of variation in aging. Based on our studies here in the United States, comparing them with those in Poland, that the availability of key resources, perhaps calories, can temper the effect of oxidative stress on aging in human females, which supports many of the ideas that have been put forward by Grajna herself and others. Um, and then the other thing that I've actually thrown into uh, previous grant proposals within the United States, if we are able to submit any proposals anytime soon, um, is the fact that um, this research actually informs health education on the effects of childbearing, especially in less resourced and marginalized communities. I think that's really important. I think, and I put this little reminder here for me that a paper that Grashina and her colleagues we contributed to on this paper in the Lancet on how human reproduction and health can be informed by an evolutionary perspective. And in that particular paper, there was a lot of discussion on the important contributions for understanding things like reproductive cancers. But I think that can be broadened, I think, in terms of how childbearing um, uh, can affect sort of long-term health prospects and how informed reproductive health would be beneficial for women's reproductive health. And then finally, the sources of aging in response to reproductive effort are less clear in human males, um, which leads to my final slide here, and that is, what next? Um, I think the influences of specific aspects of female reproductive effort on oxidative stress and aging are important. If we can tease out those that are related to gestation, lactation, and menstrual function, those are important. Um, a shout out to Becca and some of her research that she's doing right now. The influences of ovarian steroids on oxidative stress. There is some papers that suggest that estrogens, particularly estradiol, can be protective against oxidative stress, but that literature is kind of mixed. I think an energetic perspective would kind of add some clarity. And then some of the more exciting things that are just over the horizon about the estrobolome by Becca would be really, really interesting here. And then finally, pivoting back for a moment to males, I think we need to reassess how reproductive effort is measured in human males. Um, Sexual dimorphism, gamete production, paternal investment, these are all things that I think need to be woven together in a much more comprehensive fashion to understand male reproductive effort. And then perhaps maybe some of this will be a little clearer. So that is it. That is what I have. I want to really, really thank a lot of people who I'm looking at some familiar names here on the screen. Um, but in terms of resources, really want to thank the Polish National Science Center, Yale University, the National Science Foundation, um, and a number of other people within my lab and down here among the Shuar. But a really special thanks to Gražian and the research team, and especially to Anya, Ula, and Magda, who were really fun to have in the lab. I learned so much from them. And I can't wait to have more researchers come into the lab and strengthen this bond we have between Jagiellonian and Yale. So thank you so much. I appreciate you listening,